Hello everyone, welcome to ITLS Academy. This is your mentor Dr. Shamaita Basu. In this session, we will be talking about part 2 of the topic microbial pathogenesis. So we will be starting about now this is something I think I have already talked about in the previous slide about the different virulence factors and previous slide as in previous session and how they will be responsible for contracting the disease in humans. And this is also something I have talked about in the previous session only, about the different adhesive virulence factors that are present in the different bacteria, and what are the type of infections that they are responsible, or diseases that they are responsible to produce. Now, so in this session, we will begin with the different portals of entry. Okay, so portals of entry, starting with the number one, the first portal of entry will be the mucous membrane. Okay, so there are some common diseases which will be contracted via the respiratory tract, and they will include diseases like common cold, tuberculosis, whooping cough, etc. So this is basically a nice diagram of the upper respiratory system, because most of the bacterial infections like respiratory bacterial infections are responsible to infect the upper respiratory tract. Okay, so see, these are the different parts that we have. So we have the nasal cavity, we have the middle ear, the auditory tube we have, then we have the tonsils, the pharynx, okay, the esophagus, larynx is there, okay, windpipe is also there. Okay, so, in the respiratory tract, the microbes will be inhaled into the mouth or the nose in droplets of moisture or in the form of dust particles. And that is the easiest and most frequently traveled portal of entry. So, secondly, we have the gastrointestinal tract. Okay, so see again, this is a nice diagram of the gastrointestinal tract, as we can see. So, these are the various parts. We have the oral cavity, then we have the tongue, then we have the teeth, then we have the different salivary glands. Parotid salivary gland is marked over here. Then we have pharynx, we have esophagus, this is the liver. Then we have the gallbladder, okay, the duodenum, the pancreas, which is the leaf-like structure. Then we have the small intestine, the large intestine, and the anus. So the microbes will be gaining entrance through contaminated food and water, or the fingers and hands. And most of the microbes that enter the GI tract will be destroyed by HCl and enzymes of stomach, or bile and enzymes of the small intestine. Okay, now this is something very important, because the pH of the stomach is acidic. I think this is again something I have talked about in the previous sessions. Why acidic? Due to the presence of HCl, that is hydrochloric acid. Okay, so it is responsible for causing a pH of around 2 to 2.5, which is highly acidic, and that pH is actually very much unfavorable for most of the bacteria. Okay, but still there are some of the bacteria which can thrive in this acidic pH also, and they will be mainly responsible for causing the gastrointestinal tract infections in the body. Now, there are some common diseases which are contracted via the GI tract or the gastrointestinal tract. What are they? Number one, we have salmonellosis, which is caused by Salmonella species. Then we have shigellosis, which is caused by Shigella. Then we have cholera, which is caused by Vibrio cholera. Then we have ulcers, which can be caused by Helicobacter pylori. Then we have botulism, which will be caused by Clostridium botulinum. So these are the some of the common diseases which are responsible for causing infections of the, or diseases of the GI tract. Okay, so next we have the fecal oral diseases. So these pathogens which will be responsible for causing fecal oral diseases, they will enter the GI tract at one end and exit at the other end. So GI tract ka one end and other end matlab obviously it will enter through the mouth, and exit through the anus. Okay, so they will spread by contaminated hands and fingers and or through contaminated food and water. So contaminated hands and fingers are very much probable in case of children, isn't it? Because they obviously, moreover, ek to they are children, so they do not have the, the understanding or the sense of hygiene. And secondly, they are always doing something or the other. They are always active. They are always touching dirty things. Whatever they get, they have a tendency to grab it and put it inside the mouth. Okay, so like spreading of diseases through contaminated hands and fingers will be most probable via through children, in case of children, and also in case of elderly person. In case of elderly person also, it's the same thing, that they have a reduced sense of understanding, and they have the tendency, because it's told that when a person becomes older, becomes aged, he or she gets back to their childhood days, because they start behaving like child again. Okay, and contaminated food and water, जिसका ideal example होगा typhoid and cholera, which I have already talked about. And also poor personal hygiene, which is mainly seen in poor children and in illiterate children or illiterate populations. Poor, and those places where proper sanitation practices are not there. Okay, proper hygiene is not maintained due to lack of infrastructure, due to lack of proper sanitation. So in those places, this poor personal hygiene can also be uh, like a big factor for fecal oral diseases. Okay, then we have conjunctiva. Conjunctiva, mucous membrane of conjunctiva can also be a portal of entry for the microorganisms. So this is basically a diagram where an infected conjunctiva is zoomed in and focused. Okay, so the mucous membrane of the conjunctiva will cover the eyeball and it will line the eyelid. So it's a immense means of protection, immense means of protecting the eye from any kind of infection. Okay, and like the bacteria responsible for infecting or responsible for entering inside the body through the mucous membrane of the conjunctiva will be chlamydia trachomatis. Okay, and the disease caused will be known as trachoma. Second portal of entry will be the skin. Okay, so over here a diagram has been I have given, which uh, showcases the different skin manifestations which happens in case of different bacterial diseases. So here we can see red rashes or red blisters has occurred on the skin of the infected person. So skin, as we all know, it is the largest organ of the body. Okay, so largest organ of the body means what? It is also the largest, so it can also be the largest source of infection in the body, isn't it? Because it is something which is present all over the body. So from any point of the body via skin, the infectious agent can enter. When unbroken, it is an effective barrier for most of the microorganisms. Okay. Some microbes can gain entrance through openings in the skin like hair follicles and sweat glands and wound. So hair follicles and sweat glands are something which will be there in each and every person, irrespective of irrespective of whether that person is suffering from any cut or wound or abrasion, isn't it? So hair follicles and sweat glands are also sebaceous glands which secretes sebum. These are very good sources of entry of bacteria or microorganisms through the skin, and also they can enter via wound. Okay, wound means it can be a cut, it can be a surgical opening, it can be a dressing. Okay, it can be any type of abrasion, it can be any type of pus or secretion. So these are all ways and means of bacteria or microorganisms to enter via the skin inside the body. So here we can see it's a nice diagram of the different layers of the or the different components of the skin. So we all know that skin has three layers. The outermost is the epidermis, as we can see over here. These are the hair follicles that are coming out from the skin. Okay, like these are the hairs and these are the hair follicles. These are the hair follicles. Okay, so this is the epidermis. From the epidermis, the hair follicles will come out. Then the middlemost layer of the skin will be the dermis. So from the middlemost layer only we can see that the blood vessels or the blood supply are starting, isn't it, from the middle layer of the skin. And the innermost layer of the skin will be the hypodermis, where the fat layer is starting from. See, yellow colored means is fat. Anything that is denoted with red is blood, and anything that is denoted with yellow is fat. Okay, so in the hypodermis we have the fat layer. Okay, and in the diagram below, some of the physical barriers are shown. See, so the physical barriers will prevent approach of and deny access to pathogens. So skin itself is a physical barrier, largest physical barrier of the body, since it is the largest organ of the body. Okay, so among the skin, some of the additional physical barriers will be the duct of the endocrine sweat gland. So this is the duct of the sweat gland, which is responsible for releasing sweat. Again, secretion of sweat is itself a defense mechanism, because through the sweat, many bacteria, many microorganisms can be swept off via the sweat. Okay, so sweat secretion itself is a defensive mechanism of the immune system of the body. Then we have the hair coming out from the hair follicles, and we also have various other secretions like sebum, which is coming from the sebaceous glands. Okay, and all these hair follicles, these endocrine glands, they are actually coming out from the epithelium layer of the skin. Now coming to the third portal of entry after skin, that is the parenteral route. Again, something I have talked about in the previous session. Parenteral means via injection, via cut, or via any kind of opening where the bacteria or the microorganism will be able to deposit itself. Okay. So parenteral route will be where the microorganisms are deposited into the tissues below the skin or the mucous membranes. Below the skin means after the layer of the hypodermis, after the innermost layer of the skin. Okay, means after the fat layer. Okay. So it can be through punctures and scratches. Punctures will be ideal example of the intravenous injections, isn't it, where it's directly punched inside the skin, into the skin, to the muscles. And scratches, any kind of scratch on the skin, it can be via injection, it can be via bites, it can be via surgery. Because surgery, though, obviously, because it's an opening, it's an incision. Any kind of surgery means some or the other kind of incision is being made. It can be a major incision, it can be a minor incision, but there is an incision, there is an opening, so always there is a chance of infection. Okay, now coming to the preferred portal of entry. So in case of streptococcus pneumonia, which is responsible to cause pneumonia, that is a lung infection which can lead to accumulation of water, accumulation of fluid inside the lungs, and it can also lead to respiratory distress and finally choking, that is stoppage of breathing. The person can choke to death. Okay, so this streptococcus pneumonia will be inserted or will be getting access inside the body via inhalation. Inhalation mainly through aerosols. And it can also enter the GI tract, but in that case it will cause no disease. Obviously, because each and every bacteria will not be able to cause disease in each and every part of the body. Isn't it? It will vary from type to type, from what kind of species it is. Okay, so streptococcus pneumonia means it will only be able to cause pneumonia. That means in the lungs. So if it enters via the GI tract, it will be of no use. It will not be able to cause any kind of disease. Next we have Salmonella typhi. In case of Salmonella typhi, it's just the opposite way around, isn't it? If it enters via the inhalation or if it enters via the skin, it will not be able to cause any disease. But if it enters via the GI tract, then only it will be able to cause its specific disease, that is typhoid fever. Salmonella typhi is responsible for causing typhoid. Another important strain is also there of Salmonella, which is also responsible for causing typhoid, that is Salmonella paratyphi. Okay, but only if it enters via the GI tract. What is colonization? I think something I have already talked about in the previous session. So it is another critical point in the production of disease, and it refers to the growth of the microorganism on epithelial surfaces such as skin or mucous membranes or other host tissue. Which host tissue? Again, it will depend on what type of bacteria it is and where the bacteria is able to cause the disease. So colonization means when the bacteria is replicating and multiplying. So just by entering inside the body, it will not be able to cause disease. In order to cause a disease or infection, it needs to multiply and replicate inside the body. That is what is colonization, means forming colony. Colony of many bacterial cells together. Okay. Now a very important topic, that is number of invading microbes, which is again a very predetermining factor of causing disease or infection. Okay. So it will depend on how many number of microorganisms are actually invading or are actually causing invasion inside your body. Okay, so depending on that, two important terminologies are there. One is LD50 and one is ID50. Now first let's talk about LD50. LD50 stands for lethal dose 50. Okay, so 50 means half. Isn't it? 50% of population means half of the population. So if there are 100 bacteria, 50% means 50. Isn't it? So lethal means what? Lethal means death or killing. So lethal dose of a microbe toxin which will be able to kill 50% of experimentally inoculated test animal is the LD50 dose. Okay, so say for example the LD50 dose of Mycobacterium tuberculosis is 10. Okay, that means at a dose of 10, Mycobacterium tuberculosis will be able to kill 50% of the population or 50% of the host bodies where it is infecting. Host bodies means obviously human in case of Mycobacterium tuberculosis. Okay, so that is LD50. And what is ID50? ID stands for infectious dose. So infectious dose means able to cause infection or able to cause disease, not able to cause death. Okay, or not able to kill. So ID50 means the dose where the microorganism will be able to kill 50% of the number of test animals or experimental test animals where it is getting inoculated. Inoculated means getting introduced via experimental conditions. Okay, that is inoculation. So LD50 means 50% of the dose at which it will be killing 50% of the test animals, and ID50 will be dose at which it will be able to cause disease or cause infection in 50% of the test animals. Okay, so I hope it's clear the concept of LD50 and ID50. Moving forward, how do bacterial pathogens penetrate the host defenses? So I've given a nice diagram over here. So you can see this is the body of the microbe. So it can be a bacteria, it can be a fungi, it can be a parasite. Let's just imagine it's a bacteria. Because among the microorganisms, among the different types of microorganisms, bacteria form the major type of microorganism. Okay, 70 to 80% of the microorganisms are made up by bacteria only. The remaining 30% will belong to virus, parasite, fungi, etc. Okay, so majority wins. Majority is bacteria. So we will be talking about mainly, we will be focusing mainly on bacteria. So say for example, this is the body of a bacteria, say Escherichia coli or Mycobacterium tuberculosis. Okay, so it is having different ligands which are located on the surface of the cell of the bacteria, and these ligands will be producing chemicals called adhesins, which will go and react with the specific receptors present on the host cell surface. So that means a proper interaction or a proper binding or attachment with the host cell surface, and that is enough in order to penetrate inside the cell of the host. Okay, so adherence will be almost all pathogens will be having a means to attach to the host tissue, and the binding sites can be adhesins or ligands. Okay, so some bacterial cells are there which use fimbriae in order to adhere. So see, this is a typical structure of a fimbriae that we have. See these appendages, these extensions, finger-like or thread-like projections, they are the fimbriae. And this is basically the microscopic view where they are zooming in on the fimbriae. So the fimbriae can play a role in tissue tropism. What is tissue tropism? Again, something I have talked about in the previous sessions or previous topics. So tissue tropism means which will determine what kind or what type of tissue the bacteria or the virus will evade or evade. It's what type of tissue the bacteria will invade or attack. That is the tissue tropism, and that will again vary from one type of microorganism, one type of bacteria to the other. Okay. Example will be attachment of candida to the vaginal epithelial cells. What is candida? Candida albicans, it's a fungus responsible for causing fungal infections mainly in the vagina of women. Okay. Again, a diagram I have given over here, which again shows the different fimbriae. So this is a colorful diagram. That was a black and white one. So examples of bacteria which express this fimbriae and adhesins will include Neisseria gonorrhea. It will include enterotoxigenic E. coli, which is abbreviated as ETEC. Enterotoxigenic means it will uh, invade or it will infect the uh, gut. Okay. Okay. And we also have Bordetella pertussis. Now talking about bacterial toxins. Now what is toxin? It is any substance that is poisonous to any other organism. And bacterial toxins can either be exotoxin or endotoxin. So exo means it's produced outside the cell. Okay. So exotoxins will be soluble substances which will be secreted into host tissues, and they are mostly polypeptides which are more powerful and specific than endotoxins, and they can be denatured by heat to form toxoids. Uh, so the exotoxins are the ones which can be denatured by heat to form the toxoids. Now several gram-positive bacteria are there which produce exotoxins, like we have Clostridium, we have Bacillus, we have Staphylococcus, we have Streptococcus. And several gram-negative organisms are also there which are responsible for producing exotoxins, like we have Vibrio cholera and we have toxigenic E. coli. Now many exotoxins are of the AB type. So the B subunit will bind to the surface membrane of the cell, where the A subunit will be the business end. So two subunits will be there which will be making up the exotoxin, among which the B subunit will go and bind to the surface membrane of the cell. Okay. So next we have endotoxins after exotoxins. So endotoxins will be forming a part of the cell wall, because see, endo means inside. So those toxins which will be formed inside the cell of the bacteria will be the endotoxins. Now what is LPS? LPS is a part of the cell wall of the bacteria, that is lipopolysaccharide, as I already mentioned in the previous slide. So it is a part of the cell wall present particularly in gram-negative organisms, and these endotoxins will be released into the host tissues when the bacteria die or divide. Endotoxins have non-specific effects such as fever or a sudden drop in blood pressure. They also cause tissue damage in diseases such as typhoid fever and epidemic meningitis. Okay, so typhoid fever and epidemic meningitis are the particular diseases where endotoxins are produced. So that means Salmonella typhi, responsible for causing typhoid fever, and meningitis means meninges, any kind of bacteria which is responsible for inflamming the meninges will be responsible for causing meningitis. So mainly gram-negative organisms are the group of bacteria. Gram-negative bacteria are the bacteria which are responsible for producing the endotoxins, and the endotoxins are actually not that much harmful as the exotoxins. Exotoxins are much more harmful, and they are produced by both gram-positive and gram-negative organisms, while endotoxins are less harmful and they are produced by the gram-negative organisms only, and they are responsible for producing non-specific symptoms like fever, not particular specific pathogenic symptoms. Okay. Okay, now listing the important differences between endotoxins and exotoxins, again something very important. So the endotoxins will be forming an integral part of the cell wall, that is LPS. Okay, they will be formed as a part of the LPS, which is in turn an integral part of the cell wall of the bacteria. Okay, and exotoxins will be released from the cell of the bacteria before or after lysis of the bacterial cell. Second important difference is endotoxin is LPS, means lipopolysaccharide in nature, and lipid A will be toxic in nature, while exotoxins will be protein in nature. Third important difference is endotoxins are heat stable while exotoxins are heat labile. Means endotoxins will be tolerant to heat. They can tolerate heat. They can tolerate higher temperatures. But exotoxins, they will not be able to tolerate heat or tolerate higher temperature. Hence heat labile. Labile means sensitive. They are sensitive to heat. Okay. Fourth important difference is that endotoxins will be antigenic, but their immunogenicity will be questionable, while exotoxins will be both antigenic as well as immunogenic. Now, there is a very subtle difference between antigenicity and immunogenicity. What is that? Antigenicity means ability to act as an antigen. That is antigenicity, which is present both in case of endo and exotoxin. But immunogenicity is the ability to generate the immune response, means the ability to generate induction of production of antibody, which is absent in case of endotoxins. And that is the reason why endotoxins are not that much powerful or that much harmful like the exotoxins, because they are not having the immunogenicity. Not having, or at least it is questionable, as they have mentioned over here. Questionable means it's doubtful whether they are having a profound immunogenicity or not. Okay, but in case of exotoxins, they are not only having antigenicity but also immunogenicity. Another important difference, number 5, from endotoxins toxoids will not be produced. Okay, because they are heat stable, so they will not be denatured, and hence they will not be able to produce, or they will not produce rather, toxoids. But exotoxins, they are heat labile, they are sensitive to heat, so once they are heated, they will be denatured and they will be producing toxoids. Again, one of the reasons why endotoxins are more effective or more powerful in causing disease than endotoxins. Next important difference, point number 6, endotoxins will have many effect on host, while exotoxins will be specific in effect on host. Means endotoxins will be having non-specific symptoms or non-specific features they will produce, like fever. Fever is something which is not specific, isn't it? Fever can arise due to any condition. Fever can arise due to nausea, fever can arise due to diarrhea, fever can arise due to dehydration, fever can also arise if a person is suffering from cancer, fever can also arise if a person is suffering from HIV or COVID. So it is not something specific, it is a very generalized symptom. Okay, and that is what is caused by the endotoxins. But the exotoxins will be producing very specific effects. Okay, which will be specific to the type of bacteria or to the type of species of bacteria. Okay, and the last difference will be that endotoxins will be produced only by gram-negative organisms, while exotoxins will be produced by both gram-positive and gram-negative organisms. And again, that is the reason why gram-positive organisms are known to be more toxic and more pathogenic than gram-negative organisms. Okay, so I think that's it. And we also have have a table which actually enlists the different enzymes and toxins that can harm eukaryotic cells, means mainly the humans. Okay, so many lists are given over here. I will just talk about some of the important enzymes. So we have coagulase, again something I've talked about before, which is produced by Staphylococcus aureus. It will help to produce blood clot inside the patient, and after releasing that blood clot, along with the blood clot, the pathogen will be able to come outside from the blood vessel. Okay, then another important is hemolysin, which is produced by Staphylococcus, Escherichia coli and Clostridium species. Hemolysin hemo means blood, means the erythrocytes, RBCs, and lysin means lyse. So they will just simply go and lyse the RBCs of the host. Hyaluronidase will be produced by different groups of streptococci, group A, B, C and G, and also staphylococci. They will be hydrolyzing the hyaluronic acid, which will be helping to digest the cell wall, and hence they can cell membrane, and hence they can enter through the cell membrane. Okay, then there are proteins like lecithinase, okay, phospholipase produced by Clostridium species. Protein A is there. Okay, then streptokinase is there. Okay, which will bind to plasminogen, and it will inactivate the production of and hence it will digest the blood clots and release the blood clot. and thereby along with the blood clot they will come out through the blood vessels. Okay, so I think that's it. So we actually completed with microbial pathogenesis, and then we will move on to the next topic of medical biotechnology. Till then, thank you, goodbye. Please like and share and subscribe ITLS.